that, I'd like to introduce our final speaker of the first session. If she's here, I see her here. Hi, Sophia. Great to see you.
Hi.
So
Thank you.
it's my honor to introduce Sophia Liu, who is joining us from Harvard University, soon to actually be joining the Reagan Institute. And her talk will be titled Spatial Maps of T-cell receptors and transcriptomes reveal distinct immune niches and interactions in adaptive immune response. Thank you for joining us, Sophia, and please take it away.
Thank you so much, Payam. And of course, to all the organizers for the opportunity to speak and putting together this really cool conference. So yeah, I'm a graduate student, and today I'll be talking about our work spatially mapping T-cell receptors and transcriptomes. This project has been a collaboration with Brian and Shu Chang. We're a part of Ken and Kathy's groups at the Dana-Farber Cancer Institute. So to better understand the cell interactions that define immune systems, we need spatial methods. And I think of the important questions relating to T-cell interactions as falling into two categories: developmental and pathological. On the developmental side, we're interested in understanding how the T-cell receptor repertoire develops and organizes in the thymus and other lymphoid organs and resident tissues. On the pathological side, you can think of this very simple example. So here you have some T-cell clones or T-cells with the same T-cell receptor that are much better at infiltrating into a diseased region of tissue than others. And characterizing how these T-cell clones are distributed in different pathological conditions, infection, autoimmunity, cancer, and how they might be interacting with other T-cells and other cells during the immune response would help us better understand immunity and develop better immunotherapies. So to be able to answer those questions, we developed slide TCR-seq, which links clonotype, or the T-cell receptor sequence, spatial location, and the cell transcriptional state. So we started by looking at existing spatial transcriptomic methods, and I'll explain slide-seq in detail, but the general principles apply across all spatial transcriptomic methods. So slide-seq uses these 10-micron beads. That's about the size of a single cell. And on this bead, there are many of these single-stranded DNA oligos. At the end of each DNA oligo is a poly T-tail, which can capture the poly A-tail of mRNA. Each oligo also contains this spatial bead barcode. And this allows us to map each barcode to a real XY location when we take these beads and glue them onto a slide and then image them and sequence using in situ sequencing. We can then take 10-micron sections of tissue and overlay them onto these arrays. And as the tissue melts onto the beads, the mRNA is then captured and can be prepared for downstream whole transcriptome sequencing. So after sequencing, we can then assign each mRNA to a spatial location. And these slides are 3 millimeters in diameter. So for the rest of the presentation, that's the area of tissue that we're looking at. Unfortunately, these methods aren't easily applied to study TCR sequences because TCRs are long and pretty lowly expressed. And so to overcome low expression, we add this additional targeted PCR step to enrich the TCR sequences. To overcome the sequencing issues, we can then sequence in from this targeted primer site to sequence the CBR3 variable region that defines the TCR, or we can perform long-read sequencing on a nanopore. So after doing these experiments, we can then computationally reconstruct each location, identify the TCR if there's a T-cell present, and assign other cell types and states. And I'll be talking about alpha and beta TCRs throughout the talk, but you can adapt this to any species or T-cell type as long as the sequences of the B regions are known. So we then wanted to see what questions we can now answer about human immunology. And all the samples from now onwards will be human. And when it comes to spatial niches, there are 4 types of questions that slide TCR sequencing can tackle. The first is in identifying these distinct immune niches. So regions of tissues where there might be unique pockets of T-cell clones that are spatially segregated from one another. The second is whether or not T-cell clones vary in their extent of tumor infiltration, which connects back to our initial motivation for building this tool. The third question is, how do cell states spatially vary between T-cells and tissues? And is this variation consistent between T-cell clones? You can imagine that we have many T-cell states in this diseased tissue, but how does the cell state change as a function of infiltration? And last, we also begin to characterize the unknown function of tertiary lymphoid structures. For the sake of time, I'm going to talk about these 2 stories. how we can identify these immune niches and how cell states vary between T cells and tissues. But this is recently published, so I'm happy to email the PDF to anybody, or it's online. So a great tissue to ask the question of whether or not we can identify these spatially segregated T cell repertoires is the lymph node. And we got a sample from a great collaborator and pathologist, Vignesh Shanmugam, of a reactive human lymph node. So here's this visualization of the slide TCR-seq array plotted with the cell type assignments. And each point here is one of those beads. You can see that there are these regions that are rich in B cells, or the germinal centers. And germinal centers are sites where T follicular helper cells, or TFH cells, can interact with B cells and promote somatic hypermutation. We can confirm that these are germinal centers by CXCR5 and BCL6 expression. And we see that there are many T cells throughout this section of lymph node. So by using the presence of CXCR5 and BCL6, we can then define these germinal centers as blue in these niches, and dichotomize all beads as blue, germinal center, or orange, non-germinal center. And in black, we have the TCR variable sequences that we identify plotted. So we can then consider two models of how these T cells are traveling throughout this tissue. The first is that T cells can travel throughout the lymph node freely. And the second is that T cells, once they enter intergerminal centers, are no longer able to travel freely across the lymph node and are confined to the GCs. So the question then becomes, how can we differentiate between these two dynamic models? Because we're observing a very static, steady state. We can't do these beautiful intravital microscopy experiments from Arbella Victoria's lab and Michel Nussensweig's lab in these human samples. Well, we can start by looking at the number of clones that are shared in the blue versus the non-GC orange regions. And we see that there are actually many clones that are shared between the two. This is consistent with our knowledge that T cells can migrate in from non-GC to GC regions. But what does this mean in the context of these two models, the random movement versus the spatial segregation? So we can actually test which of these two models is correct by randomly shuffling the locations of these T cell clones 1,000 times and recording what the random model would expect for the number of shared clones between these GC and non-GC regions. We can then compare what we're observing to this random model. And as you can see, the number of overlapping clones is significantly lower, which supports model 2. And this is biology that we're familiar with. Once T cells migrate into GCs, they become TFH cells and generally don't leave. As for new biology, though, what about the distributions of T cell clones between two germinal centers, which is not as well characterized? Do two separate GCs contain random distributions of T cell clones, or do they share some T cell clones but maintain largely distinct niches? And answering this question helps us understand how our immune systems generate B cell diversity. Is the process completely stochastic, as in model 1, which would suggest that we get diverse B cells because of contacts with random TFH cells? Or do we maintain these spatially distinct niches, where even these adjacent GCs contain very different TFH cells? So we can perform a very similar test where we shuffle the spatial locations and observe how many clones are shared between these two germinal centers. What we see is that the amount of sharing is much lower than expected from a random model. So you can imagine more broadly applying this sort of analysis to T cell niches in different human tissues, in the gut and the skin and so on, especially where it's difficult to perform intravital imaging to better understand the dynamics of T cell movement. So one of the more powerful aspects of spatial biology is being able to study how cells are interacting with one another. And there might be effects on cells adjacent to T cells that vary depending on the clonality of the T cell itself. So we identified a great sample to demonstrate this sort of analysis in, a metastatic melanoma sample. And the sample notably has these two tumor lobes shown here in blue and orange. They're separated by a fibrous septum, and the sample overall had a very high amount of immune infiltrate. So we took many regions of this tumor, and we then wondered if there were T cell clones that had a bias between the two tumor lobes. We found that this one clone was much more highly expressed in lobe 2. And because we have the spatial data, we can visualize this. And so we can look at where all the TCRs are. You can see there are many between the two tumor lobes. And in the right column, you can see that this one clone is really biased for this orange lobe of tumor. So what makes this clone special? We can do something, we can do something similar to what you would do in a single cell experiment, which is look at differentially expressed genes for that clone. We find that this clone expresses granzyme B and STAT3 much more highly than other T cell clones. suggesting that it uniquely has this cytotoxic response. But what about new questions about how this clone might be interacting with other cell types? So we can look at monocytes that are next to this clone and compare them against all of the monocytes next to all of the other T cell clones. What we find is that the monocytes next to this clone have much higher CXCL10 expression, which is a signal used to recruit T cells. What that means is either this clone has a unique effect on monocytes to produce CXCL10, or this clone can respond uniquely to CXCL10. And we do see elevated interferon gamma expression, supporting that this clone is uniquely causing monocytes to express CXCL10. As for tumor cells, we can perform a similar analysis. And so we find that the tumor cells that are next to all of the other clones have much higher expression of MGST1, which is recently identified as a repressor of ferroptosis. It's also been shown that CD8 T cells can regulate ferroptosis during cancer immunotherapy. And so as for the direction of this interaction, is it that the tumor cells are recruiting this T cell clone because they have lower expression of MGST1? Or is the clone repressing tumor expression of MGST1? The literature supports that this specific clone could be regulating ferroptosis in the lobe of tumor that it's in. And so all in all, I think this sort of approach has opened a new way of thinking about tumor and immune cell interactions. The reality is that these phenotypes that we observe very broadly, tumor infiltration, induced tumor cell death, can come from interactions that actually change and vary a lot depending on their location and the clonality of the T cells involved. And so in the long term, these differences might contribute to the generation of therapeutic resistance over time. So it's important that we keep studying cell-cell interactions to understand how the immune system is working at a more granular level. And so I've talked to you about how slide TCR-seq can look at immune niches and look at immune cell heterogeneity in tissues. And we're now working on extending this sort of study to include B-cell receptors and also look at host viral interactions. One thing that I'm particularly excited about is we've started applying this tool to study T-cell changes in the thymus over aging, because thymic involution is really fascinating. So hopefully I'll be invited back next year to share some of those results. And so I'd like to end by thanking everyone that I've gotten to work with, especially my advisor, Faye, who's been a wonderful friend and mentor throughout my graduate school experience, and especially as I'm moving forward to start my own lab. And I'm happy to answer any questions that you might have.
Thank you, Sophia. That was really a phenomenal talk describing a really paradigm shifting tool. So thank you for sharing that with us, and very excited to see where this research takes you next. And it's perfect timing too, as we have several minutes to have a discussion before moving on to Dr. Ardis's talk next. So there is one question so far from the audience that I can see, and this is more a technical question, wondering if it's more difficult to perform this method depending on the tissue. If you have any comments on that.
Yeah, that's actually a very good question. So there's a lot of variability. The tool was originally optimized for brain, which is like, it just wants, the brain, if you ever handle it, just wants to explode. I know Dr. Becker was talking about this, where it's hard